Well, welcome back to part 2 of our lesson on osmosis. And in this part, we're going to be looking at osmosis as happening in cells. Now, I mentioned the last time that osmosis can take place across the partially permeable cell membrane. And I left you with this question of which solution you would store red blood cells in pure water or solution with some salt in it. Now, you might have thought that pure water was a good idea because after all, pure water is so harmless and so good for us. But really, the answer is that you should store it in a solution with some salt in it. Now, why is that? Why is it not a good idea to put red blood cells in pure water? Well, we're going to look at osmosis to find out. Alright, in this slide, we'll be looking at what happens to animal cells like red blood cells when you place them into different types of solutions. So the first thing you probably realized was these three red words up here that look kind of new. Hypertonic, isotonic, and hypotonic. Now what does that mean anyway? Well, don't be afraid. Really, it's something that's related to what you have already known. And I'll show you on the next slide. Now remember how we talked about these two solutions, right? 5% solution, we'll call it A, and 15% solution, we'll call it B. And we know that A has a higher water potential, right? You can see that. So we say that A is actually hypotonic to B. A has a higher water potential than B. So we say that it is hypotonic to B. Now on the other hand, we can say that B is hypertonic to A. Alright, B has a lower water potential compared to A, so it is hypertonic to A. So that's how we use these words hypo and hypertonic. Now isotonic means that the two solutions have the same concentration. So for example, if we had a 5% compared to a 5% solution, then we'll say that the two of them are isotonic to one another. Sounds like your isotonic drinks. But basically, the isotonic drinks that you drink have a concentration of water that is similar to your body fluids. That's why we call it isotonic drink. Alright, so get your head around hypertonic, isotonic, and hypotonic. Alright, let's come back to our table here. So if you place an animal cell, like a red blood cell, into a hypertonic solution, well, that means that the water potential of the solution is lower, okay, compared to inside the cell, compared to the cytoplasm of the cell, which has a higher water potential. Then in which direction will water molecules move? Okay, you're correct to say that the water molecules would move out of the cell by osmosis, and this results in the cell shriveling up in a process known as crenation. Okay, not cremation, but crenation. Now, on the other hand, if you place an animal cell into a solution that is hypotonic, meaning that now the water potential in the solution is higher compared to a lower water potential of the cytoplasm, then in which direction will water move? Well, you're correct to say that water molecules will move into the cell, right, from higher water potential to lower water potential by osmosis. And what this results in is that the red blood cell, or the animal cell more generally, okay, so much water goes into it that it bursts. Alright, so much water moves into the cell that it bursts. And another word for bursting is lysing, lysis. Okay, it lyses. Now finally, what happens when you place an animal cell or red blood cell into an isotonic solution? That means that the water potential of the solution is the same, right? It's the same outside as it is in the cytoplasm. Equal. Well, in this case, there is no osmosis. Okay, there is no net movement of water. Now what you notice is that the water does move into the cell, but it also moves out of the cell. So at the end of the day, there is no net movement. Okay, that cell does not gain or lose water. and so it remains the same. So the red blood cell here is very happy. So that is why you do not store red blood cells in pure water. If you store red blood cells in pure water, they will likely burst. Right, and then you won't have anything left to donate. Now that is just one of the cool applications of osmosis to medicine. There are many more, which you can go and look up by yourself. And we're going to turn our attention to plant cells. So plant cells, from the previous topic, you know that they're slightly different from animal cells. Right, what are the main differences? You can think of some. And one of the main differences is that they have a cell wall. And we're going to see how this plays out. This is very important for what happens to them when you place them in different solutions. So let's start again with putting a plant cell into a hypertonic solution, which means that the water potential outside is lower compared to the water potential of the cell sap. So the cell sap, remember, is the fluid that is found inside the central vacuole. Okay, this has a higher water potential. Right, what happens? Which direction does water move? Right, water molecules move out of the cell sap by osmosis. And what happens here is that the plant cell becomes plasmolized. Okay, this is an interesting word that you should learn. Okay, the cell becomes plasmolized. And what exactly does that mean? So when the cell becomes plasmolized, when it undergoes plasmolysis, some things happen to it. Firstly, it decreases in size, right? Because water has moved out of the cell. Okay, you can see that the vacuole here has really decreased in size. The cell becomes flaccid, it becomes very limp, soft, right? Not firm. And the cytoplasm and the cell membrane shrink away from the cell wall. This is very interesting, very distinct. You see this line here. This is the cell membrane. And of course, the orangey part inside here, this is the cytoplasm. So you can see that the cell membrane, which is usually very near, very closely associated with the cell wall, has actually detached itself. It has shrunk away and detached itself. That's what happens when a cell becomes plasmolized. Now let's come back to our table here. So what happens then when you place plant cells into a hypotonic solution? Meaning that now the water potential outside the cell is much higher compared to the water potential in the cell sap. In which direction does water move? Right, you're right to say that because the cell sap has lower water potential, okay, water molecules now move into the cell by osmosis. And remember, for animal cells, what happened next? The animal cell would, boom, it'll burst. But does the plant cell burst? No, it expands. And it becomes turgid. Right? Turgid basically means that it is very firm and hard. Now why is that? Why is it that plant cells don't burst? Well, it's something to do with this cell wall. Right? This cell wall here prevents the plant cell from bursting. Unlike animal cells, plant cells have this cell wall that prevents them from bursting. And finally, what happens when you place plant cells into an isotonic solution where the water potential is equal to the cell sap? So the cell sap has equal water potential. Well, there is no osmosis, no net movement of water, and the cell just remains relatively unchanged. So nothing visible happens. I've said this already, but I'll say it again. Right, what prevents plant cells from bursting is that they have an inelastic cell wall. Animal cells do not have this cell wall, and so when too much water moves in by osmosis, they burst. So you might have seen this phrase, turgor pressure, inside your textbooks. So let me just explain what it is. The turgor pressure is something that is around when water moves into the vacuole of a plant. So imagine if water moves into this vacuole here. Over time, there's a pressure that is built up. You know, it's like filling up a balloon with water. And there's more and more pressure that will be built up inside the vacuole. And that is the turgor pressure. Right? Turgor pressure is this outward pressure that the vacuole exerts on the cell wall outwards. And that is the pressure that keeps the cell very firm. So water moves into the cell, turgor pressure builds up and is exerted on the cell wall so that the cell becomes firm and turgid. So what's the deal with this turgor pressure anyway? Why is it important? Well, look at this very healthy plant here. You see that it's standing firm and upright, able to receive sunlight. And that's because its cells are turgid. Right, this plant is receiving enough water, its cells are turgid and firm, and the turgor pressure in the cells keep the plant firm and upright. Now, this is especially important for plants that do not have hard tissue like bark. On the other hand, see this plant here, it has wilted. And that's because it's not getting enough water, and so its cells are plasmolized. They are flaccid. They are not firm. So the plant wilts. And if this keeps up long enough, the plant will eventually die. And before we end off, I just want to highlight that there are a few applications of osmosis. And we're going to be discussing these more in class together. So for instance, you might have heard about how snails and slugs can be killed by adding salt to them. Or you might have thought about the question that if you're stuck on a desert island, should you drink seawater to survive? And also the question that I started off at the very beginning, why is it that salt is used as a good preservative? Now, using what you've learned about osmosis, give it a think, give it a thought, and when we come back to class, let's discuss it together and understand how osmosis is applied in our lives. Alright, see you there.